Okay, so welcome along, ladies and gentlemen, to your weekly SETI seminar series. Today we're very lucky to be joined by Caitlin Contag. Caitlin has recently finished her studies at BS at USC in policy planning and management and health policy. Her interests have revolved around tuberculosis and non-tuberculosis microbacterial infectious diseases, and she's currently shopping around for MD PhD programs. She's recently been doing some work at a microbiome lab at Stanford, and she gave a recent presentation in the 100-Year Starship conference about the talk that she's going to be talking about us today. She's also interested in interactions between microbes and host organisms. And also exploiting diving and undersea techniques to look at microbes and astrobiological implications of such microbes. So please join me in welcoming Caitlin.
Hi, thanks Adrian. So I initially gave this presentation at 100-Year Starship, which is intended to bring together scientists who all have ideas about what technologies are needed for the mission to the next nearest star. And this conference is actually funded by DARPA, and that was about a month ago. And so Jill asked me to give the talk again here today. And the topic today is every body is an ark, which refers to the fact that if we go to space, none of us will be traveling alone. We are accompanied by countless microcosmonauts that will live in our noses, on our skin, in our digestive tract, between our toes, everywhere. And they're going to be joining us for that mission. And that can be a really good thing, or that can be a really bad thing. So the first question is, what is the human microbiome? And this term was actually coined in 2001 by a very brilliant biologist by the name of Lederberg in a paper called Sweet Omics. And he defined the human microbiome as the ecological community of commensal, symbiotic, and pathogenic microorganisms that literally share our body space. And I put on this slide an image of some key body sites that are colonized and examples of organisms that colonize them. But it's important to remember that by no means is this a comprehensive explanation or list of all the things that live in us and on us. There are many, many more than these, but these are some key organisms that colonize these body sites. So what does the microbiome do? This is a question that we've only really just begun to answer, and I've isolated 3 key areas that I think will be particularly important for a long-term space mission. The first thing is digestion, and microorganisms live inside of our gut, inside of our stomach, everywhere, and they help us digest our food. But they also contribute to some diseases of digestion, including Crohn's disease. Crohn's disease actually has a very particular gut microbiome associated with it, and you can transfer it from organism to organism by giving the microbiome of a mouse with Crohn's disease to a healthy mouse. In addition, inflammatory bowel disease, which is closely related, it can also be transferred by transferring the organisms that live in the gut from organism to organism. But most interestingly, that intestinal pain that's often caused by Crohn's disease and inflammatory bowel disease can be mitigated by the inclusion of a strain of lactobacillus into the gut, and it interacts with the opiate and cannabinoid receptors in your gut to mitigate some of that pain caused by the stretch. In addition, metabolism is extremely impacted by the microbiome in the body. Metabolic syndrome, the constellation of disorders that are associated often with type 2 diabetes, can actually, again, be transferred from organism to organism by transferring the microbiome. But in addition, it can be alleviated by long-term treatment with antibiotics, in mice models, of course, that can make them more susceptible to insulin and lower their body fat. Finally, immune function, which I think is the most interesting. And actually, when I was preparing this slide, it was hard to categorize these different disorders into digestion, metabolism, and immune because they're so interconnected. But the prokaryotic organisms that live in our gut have a very significant interaction with the eukaryotic cells of our own body. And that interplay can do great harm, but also can have great benefits. For example, colorectal cancer can be transferred from organism to organism. When you take mice that are particularly susceptible to colorectal cancer and house them with healthy mice, the susceptibility transfers between the organisms by transferring those organisms that are present in the gut. In addition, T cell function in the skin is incredibly driven by the organisms that we have living on our skin. When you take gnotobiotic mice that have no organisms living in them, function in their skin, their ability to fight off those skin pathogens is greatly reduced. But introducing just one organism, Staph epidermidis, can actually restore T cell functioning through increasing the production of interleukin-1 and the downstream signal MYD88, and causes the organisms to increase their immune function against the skin pathogens. Finally, atopic dermatitis, or eczema, that itchy, scaly stuff on your skin, you can actually track the flare-ups based on the level of diversity on your skin. As diversity falls, you get increased flare-ups on your skin. So this interplay is very important to maintaining a healthy skin and gut. So why is this important to space travel? And that's because traveling in space is kind of like being in the middle seat on an airplane in the far back. And that is, you're going to have a long journey, especially if you're going to the next nearest star. You're going to be in close quarters with your seatmates, and you're going to have a strange diet. What this means is you're going to have alterations in immune function, but there's also alterations in microbial physiology that are associated with microgravitational conditions. I actually included a picture of the space shuttle guys eating gummy worms to show just how unusual these conditions are going to be. So something really crazy happens to your immune system when you're in space, and that is that you become immunosuppressed. You get altered immune cell distribution and function. You get decreased capability to generate new immune cells. You have decreased mass of key immune organs like your spleen, liver, and thymus. And you have decreased expression of antioxidant genes that scavenge all of the free radicals in your body. What this means is that you have vulnerability to infection, including infection by organisms not generally considered pathogenic. And in addition, you get reactivation of latent viruses, so varicella or chickenpox or shingles, and Epstein-Barr virus, which causes mono, both reactivate in astronauts traveling into space because of their decreased immune capabilities. That's a big problem. Nobody wants that to happen. And in our bodies, we carry organisms that are what are considered moderate risk pathogens that normally live with us without damaging our body, but when we're immunocompromised, can actually become infectious and cause really serious problems. In addition, something really strange happens in microbial physiology during space travel. And this is what I think is most interesting when we look at organisms that are put into an extreme environment. Gravity is such a driving evolutionary force on Earth. When we take that away, what happens? And some very bad things happen as you consider astronauts. And the first thing that happens is that your skin flora, all those organisms that are keeping you safe and keeping your T cells functioning and signaling other T cells to come to sites of infection, those become more susceptible to antibiotics. And that means that if you have a small skin infection caused by something else, you put antibiotics on it, you're disrupting your skin flora more than you would on Earth. Unfortunately, there are also changes in antibiotic binding capabilities that result in more nasty bacteria like E. coli being more susceptible to, they're becoming more resistant to antibiotics, which means that as you're killing off the good stuff, the bad stuff can survive more, which means that you can have the bad skin pathogens take over your skin as you're killing off the good ones that keep you functioning. In addition, you get higher deletion mutation frequencies during spaceflight, which means that novel pathogenic capabilities could develop at a faster rate than they do here on Earth. You get increased production of enterotoxins and pathogenic E. coli. So the organisms that you hear about on the news infecting people as they eat contaminated spinach, those guys form thicker biofilms, they grow in thicker quantities, and they become more resistant to antibiotics. And those are the things that protect us from invasive bacteria, and they work less in space. And that's a problem for astronauts. And in addition, S. enterica, Salmonella enterica, becomes significantly more virulent when incubated in microgravitational conditions. They put the organisms on the space shuttle, they fly them up into space, they bring them back to Earth, they feed them to mice, and those mice die 2 to 3 times faster then Salmonella incubated on Earth. And they've identified some virulence factors that have to do with how they insert the proteins into the host cells, so our cells, but also how their flagella work. And so for some reason, microgravity causes these virulence factors to be upregulated. We're not really sure why, but it's a response to a novel environment. Finally, Candida albicans, the causative agent of thrush and yeast infections, becomes more pathogenic and grows in a way that is actually more damaging to tissue when incubated in microgravitational conditions. And when you're sending astronauts up into space, this is a really big concern. And this has actually been identified since the earliest space missions as a problem. So they've actually done a lot of epidemiology in space. And recently, I guess relatively recently, in the late 80s and early 90s, a guy by the name of Pearson did epidemiology in 2 years of space shuttle missions and found that there was, in fact, transfer of moderate risk pathogens that most people carry in their bodies between space shuttle flight members. And they identified this as not the biggest problem, but still something that needs to be considered, and there needs to be alterations made in the recommendations for how we send people into space in order to prevent that transfer of microbes from person to person. The Apollo missions actually had more interesting results of their epidemiology. So they tracked all the Apollo missions, and in particular 13, 17, 12, and 7 were the ones where this became a huge problem. They saw athlete's foot, and with athlete's foot, they also saw secondary infections with Pseudomonas become a huge problem on several Apollo missions, as well as outbreaks of jock itch, which is caused by the same pathogen. On Apollo 13, the astronauts came back with serious beta hemolytic streptococci infections in their throat, which was identified as an organism that was isolated from one of the astronauts' belly button before the flight. So somebody's touching their belly button, touching other people. And in addition, urinary tract infections became a huge problem on the Apollo missions and also on the space shuttle missions. So this has been identified as a problem. It's been tracked for a long time, and there have been some things done to make it a little bit better, and that's why the space shuttle missions were not as serious as the Apollo missions. So here's a graph done by the same guy, Pearson, that shows the isolation of colony-forming units of bacteria and fungus from the air. And one of the unique challenges to microgravity is that when you sneeze, those droplets go everywhere. They're all over the room. They don't settle. You can't escape them. So you're breathing them in all the time. And as you spend more and more time in the spacecraft, there's actually a dramatic increase in the number of colony-forming units or bacteria and fungus in the air. Fungus is a little bit different from bacteria because it needs water to grow, and as the air gets drier, it's less able to grow, but bacteria does show the steep increase as the flight goes on. This is a problem when you're thinking about sending people into space for a mission to Mars, which will take much more time than to the moon or just a shuttle flight, and even more time when you're doing an interstellar mission like the goal of 100-year Starship was. So this is something we have to consider very carefully when we think about sending people on a long-term flight. One of the things that they did to sort of mitigate this problem of the floating particulates in the spacecraft is that they installed these air filters. And after they came back from the flight, they looked at the air filters, and this is all the debris that they pulled out of it. Pretty gross. I don't want to be breathing that in, and neither did the astronauts. They complained about nose problems, throat problems, eye problems until these filters were installed. And this is an example of how to respond to the needs of the astronauts and how to make this a less serious problem. So there are some key opportunities that result from this problem. And the first one is that we can eliminate common pathogens. If we're sending people on a long-term space mission, we should probably think about what organisms they should carry. Should we send people with herpes? And is that going to be a problem? And I think that by limiting the pathogens that people board the spacecraft with, there can be health savings, but also the quality of life of the astronauts can be improved. The second thing that is a great opportunity is developing targeted probiotic therapies. And I'll talk about each of these a little bit later. But we can actually design a microbiome that will work with the environment that the flight will be in. So we can make sure that the astronauts have a complement of organisms that will fit their diet, but also the environment that's around them. And finally, something that I think is really cool is that we should send people in a long-term space mission with a library of organisms, not just for the health of the travelers, but also to do research while we're in space to see what microgravitational conditions do to bacteria, which can tell us something about what organisms in microgravity or gravitational conditions different than Earth do and what do they look like. So the first thing I think we need to do is eliminate common pathogens. And currently, there are screening mechanisms in place to make sure that people with latent TB, which could become tuberculosis, which could become a big problem when you're in close quarters, make sure those people are not included in the astronauts. Colonization with Clostridium difficile, which causes a really serious colitis in immunosuppressed patients or patients on long-term antibiotic therapy. That should probably disqualify a candidate. And then Salmonella enterica carriers, which are, so Salmonella has various serovars, so different types of it, and they cause typhoid fever, but they also are one of the biggest causes of bacterial contamination in food. And after all, Typhoid Kirk doesn't really have the same ring as Captain Kirk. So that's definitely something to consider. People with Salmonella probably should not be included on a long-term space mission. But there are definitely more ambiguous cases. So trichophyton is the fungus that causes athlete's foot and jock itch, and about 10 to 15% of the population has it, which means that some of you in here have it. But does that mean you should be included or discluded from the space shuttle mission? I don't know. And then Streptococcus mutans is a bacteria that's associated with dental decay. Not having dental decay on a long-term space mission would definitely reduce costs and that people wouldn't have to have as much dental care. But should that really exclude somebody from being an astronaut? And again, the question is, I don't really know, and those are things that we have to consider as we plan for a long-term space mission. And as we go through the screening process of who should be included, what should be included, there's the possibility of excluding important organisms. Like I said, this is a pretty new area of study, and we don't quite know what everything that we have does yet. And if we decide that we only want these organisms included, or we want to get rid of these organisms, and we prescribe an antibiotic course, that means that something that could really help us, we would exclude from the mix. And we're sending people into space, and there's no way to get that back. And decreased microbial diversity is, of course, associated with all these bad irritable bowel diseases or the skin problems. And so we want our astronauts to have sufficient microbial diversity. The second opportunity is to develop targeted probiotic therapies. And right now, you can go to the grocery store, you can walk down the yogurt aisle, and there's all these claims about how their proprietary strain of bacteria is going to increase your immunity, decrease diarrhea, etc. But none of them are FDA approved to actually do those things, nor have any of them shown any efficacy at all, at least the commercially available strains. That's a big problem when they're selling it to people. And one of the things that can really benefit Earthbound travelers as well as space travelers is developing these probiotic therapies that actually work. And there's actually some studies, Lawley in 2012 had a set of patients with C. difficile, again, which causes the horrible colitis, bloody diarrhea, a lot of pain. He actually treated them with a probiotic strain that he developed, which had greater efficacy than vancomycin treatment. And vancomycin is currently the standard of care for C. difficile treatment. So it can be done, but how do we get those into commercially available products? The other thing that would be really cool to do is develop an ideal gut microflora alongside the diet. The diet of any space traveler is going to be different than the diet on Earth because of the limitations of spacecraft and the limitations of food that you can bring with you. And so we need to develop a microflora that can respond to that. And actually, vegetarians have a very different complement of organisms living in their gut than people who are meat eaters do, which is an indication that we need to think about how people are going to respond to the food we feed them in space. And the other thing I think we need to do is develop the microbiome alongside the spacecraft. So we're going to know what environment they're going to be living in. We have to make sure we send them with the bacteria that can respond to that environment best. The third thing that I think is a great opportunity is to send the travelers with a library of organisms. And that means that on the go, on the fly, we can develop new therapies to respond to challenges that we might not be able to think of here on Earth, as they're going to be in flight for a year, 2 years, 5 years, 10 years, 50 years maybe. And we can also investigate the impact of microgravity on the microbiome. There's only really been 2 organisms studied extensively in microgravitational conditions, and that's the E. coli and also Salmonella. But what about the other stuff that lives inside of us? We need to know what that does too, and that's a great opportunity for space travel if we're going to think about sending people into space for this long. Additionally, with the library of organisms, we could maintain the traveler's diversity. We could prevent the monoculture that results in those irritable bowel disorders. And we can also maintain the protective microorganism barriers that are on our skin that prevent us from pathogens that come in. So it's actually it has significant immune function. Finally, reseeding. So here on Earth, every day we walk around, we shake people's hands, we touch our face, we touch our eyes, and we reseed our microbiome with other people's organisms, with novel organisms. In space, you're going to be with the same group of people for that many years, and you're not going to have the food from Earth. You have to be able to get new organisms into your body and prevent the monoculture again. And we can re-inoculate following antimicrobial therapy as well. So if somebody gets sick on space, they have a long course of antibiotics, they kill off the good stuff in their body, we can give them new stuff. But there are challenges. Like I said, this is a very new area of research, and only recently have we had the genetic tools to really understand the populations inside of us. And so there are some major limitations that we have. And the first thing is applicability to life in space. Most of the studies done on the human microbiome are done in 1G on the surface of the Earth in people who are eating normal food, not the MREs, the meals ready to eat that you'd send with them in space. And so we don't really know what that does to our microbiome. And in addition, astronauts face significant stress. What does stress do to your microbiome? Studies suggest probably not very good things. And so we have to understand what our research and our data here on Earth will tell us about the people who we would send out into a space mission. The second major challenge is that we have limitations in assays. Right now, the standard for researching the microbiome is something called 16S RNA sequencing, which looks at a highly conserved sequence of RNA. And the problem with that is that you can't see the horizontal gene transfer that takes place. So in our gut, we have organisms that transfer DNA in the form of plasmids from one individual organism to the other. And just sequencing the 16S RNA won't tell you if one organism has new genes or not. A good example of why this is important is Vibrio cholera, which is the causative agent of cholera, isn't actually infectious unless it has a phage, which is a virus that infects bacteria. It's not infectious unless it has a phage associated with it that produces the toxins or impacts the organism such that it does produce toxins. You find Vibrio cholera out in the ocean, you could go take a sample, you can find Vibrio in it, but it's not until it gets this phage that it's actually infectious. Horizontal gene transfer is a big deal when it comes to organisms. Finally, there's some sensitivity issues. We don't know if our current assays actually test for the things that we want. The other problem is that this is a relative area of novelty. Many organisms that we have living in our body haven't actually been described. And if we're sequencing their genome and then testing that against a bank of organisms that we already know, it's possible to miss things that we've never even described that aren't included in our already known bank of organisms. And that's a problem when it's such a new area, such an unknown region of our body, the thing that we live with every day. And I like to say that you don't always get what you want, but you always get what you assay for. So unless we have the right assays, we're not going to find out what's actually living in us. The other problem is that we have geographically and culturally narrowed data. Much of our data about the microbiome is centered around academic medical centers because those are the patients that are available to researchers. But we need to think about, what is a human living in Africa? What is their microbiome like? What is somebody with a totally culturally different diet from us? What is their microbiome like? Because we're going to be sending people out into space that maybe don't have the same environment and diet as the people that are generally tested here on Earth. So I suggest a handful of studies that I think have great relevance to long-term interstellar travel. And again, the impact of microgravity simulation on the mammalian microbiome. And I think this is something that is of great interest to a place like SETI, to look at what do organisms look like in areas that don't have 1G. What does that look like? How does that impact the expression of genes? How does that impact their virulence? What do organisms do when they're not exposed to Earth's gravity? The other thing I think that we should look into is hyperbaric conditions as a possibility to overcome immune dysfunction. There's some evidence that suggests that hyperbaric conditions, so increasing the pressure actually alters the way your immune system works. And if we're artificially going to be creating pressure in a spacecraft, we can probably up that pressure a little bit and think about what that does to the astronauts. I think that we definitely, more than anything, need longer and larger epidemiological studies of moderate risk pathogens in spacecraft. In the 80s and 90s, there was about a 2-year period of space shuttle flights that people studied to determine what it means for a group of astronauts to be in close quarters, and do people actually transfer organisms from person to person? And we don't know the full answer to that because these are studies taken over 12 days, 5 days. What about a year? That's very different. We need standardized metagenomic analyses with high reproducibility. Like I said before, there are limitations in the assays that we use now. We need to standardize those to make sure that you can go to India or Bangladesh, and the academic centers there are going to be using the same stuff that we're using here, so we can compare apples to apples. Finally, we need to meaningfully and beneficially alter human microbiome using probiotic therapy. Activia is not the answer for our astronauts. We need something that's actually proven to work, something that's tested and standardized, randomized, controlled trials, if we're actually going to recommend to people that they take it up into space, because weight is a big consideration. Space is a big consideration. We need things that actually work, and we need things that work for us here on Earth. Finally, there are some ethical considerations that go along with shaping the microbiomes of astronauts and taking microbiome data from astronauts. The first one is, how should microbiome data be protected? One of the big problems is that even though astronauts are public figures, they're doing public work, their medical data is still protected by HIPAA, which are the laws that regulate privacy and medical records. In the epidemiological studies, there's a case of an astronaut who has herpes that gets transferred to another astronaut. But they can't name them, they can't describe the shuttle they were on, because that would give away too much medical information. So the microbiome data is almost as personal as your genetic sequencing, and in some cases, your microbiome is actually as unique as a fingerprint. And so this needs to be protected, and the privacy of the astronauts needs to be considered as we do these studies. There's invasiveness of sampling and privacy considerations. So the major sites of the body that are colonized with bacteria and fungus are usually sites that you don't want your doctor sticking a swab in. And I think that there needs to be some consideration about how we protect sort of the dignity of people who are being assayed for their microbiome. And when you're thinking about creating a microbiome in a human being that's incredibly unique, that's designed and planned for them to go into space, there's only really one way to do that, and that is to raise them like we raise gnotobiotic mice, which is C-section, isolation chamber. That is absolutely in no way an option for long-term space travelers or anyone really. That's not a way for a human to live. And so completely shaping the microbiome of a traveler is just not going to happen. So we have to think about other ways where we can complement and modify it in a way that's not so psychologically damaging to a human. And I just want to leave you guys with this question. Should the isolation of Staphylococcus aureus from your anterior nares prevent you from participating in a brave new world? Just because we take something from you that could be pathogenic, does that mean that you're not fit to travel to the next star? I don't know. I think that's something for us all to consider. Thanks. I would love to take some questions.
I'll start the questions, Caitlin, and then I'll go around to people. So the shuttle and ISS's and Apollo flights were good for testing microgravity. What about reduced gravity, like on Mars, or perhaps if they had artificial gravity en route to Mars? Has any studies being done on just reduced gravity conditions?
Unfortunately, I have kind of an unsatisfying answer to that. And the answer to that is there have been no studies done on reduced gravity and bacteria, only microgravitational conditions. And we actually have incubators that rotate the organisms to mimic microgravity. And some studies have been done in sort of these devices, but also some studies have been done actually taking the organisms up into the space flight. So unfortunately, we don't know the impact of reduced gravity or even greater gravity than 1G on organisms. But I suspect that, like microgravity, there are some changes made.
Your question about latent TB, it's an issue of presence and absence. You're part of a generation, as well as some of the older people here, where there's this line, which only in the last decade or so has come to light, and that is we've stopped immunizing people with cowpox against smallpox. And okay, so we don't fly people with latent TB. The question I have is, there's a whole generation, especially of young people, who don't like needles. This was just in the Mercury News on Sunday. And they don't want to have what a lot of other, tetanus and a variety of other things. Should they also be, should the people who refuse these immunizations also be considered reject material for space flight as well, too?
So when you fly in the space shuttle, you're required to have all recommended immunizations. There are no exceptions for you because of the danger that an infection could pose that we normally immunize against. The problem with the vaccine strain for TB, BCG, cowpox is for smallpox. So BCG is really only effective at preventing tuberculosis in about 50% of the cases. And so it's not an effective vaccine. In addition, you're inoculating them with an organism that could come back and be a problem if you're in an immunocompromised state. So I definitely think that we need to consider carefully what vaccines are recommended. I would also like to say that my generation definitely, fear of needles or not, we're big on vaccines. So I think I don't think that fear of needles is going to prevent people from being immunized.
Hi, the original NASA plan was to send astronauts to Mars next year, 2014, but that's been canceled. Does anybody here know when this will actually happen? Have you heard anything?
I have not heard anything.
Because it's definitely what you said relates.
I think it's not this year.
We've got some more time to do more studies. It's a blessing.
Somewhere recently I read that the microbiome might have at least as many cells, if not more, in your body than there are human cells. Is that true?
Yep. So you are more bacteria than you are human.
Caitlin, since your crew is going to be geographically and culturally diverse,
Yes.
is there any expectation that after a period of time in a enclosed environment with a regimented diet, that they will come to some sort of homogeneous microbiome so that you could do the kind of thing that you were talking about with supplementing the diet with what you need to keep everybody healthy?
Yeah, Jill. So there's definitely homogeneity when even you house mice together. So housing mice together causes them to get this uniform microbiome. And I think that you're going to see when people are in close quarters for a long period of time, you may not eat each other's poop like mice do, which definitely contributes to the homogeneity. We are definitely going to be, they're going to be in close contact, and they're going to eventually, yes, I think, have a very similar microbiome, even if they come from diverse geographic regions, which might be a good thing because I think that there's a process of natural selection that the organisms that do best in this initially heterogeneous group are going to be the ones that eventually form the group that is the ultimate homogeneous microbiome of the travelers. So I think that's a really interesting point.
2 questions, if I may.
Yes, of course.
So you've talked about effects of stress and closed quarters and so on, in addition to effects of microgravity. Has there been work with, submariners who are under the water for 6 months, or people in Antarctica for winter and so on, to try to separate out what is stress and lots of physical activity and what is, and crowding, and what is gravity? And then you also talked about effects of pressure. How much do you have to up the pressure to start having immune system changes? Because you don't want to give the astronauts the bends or set things on fire.
Okay, so to answer your first question, I think that you're right in thinking that deep sea submarines and sort of the Antarctic travelers would be really good model communities for epidemiological studies of what happens to the microbiome in space. Unfortunately, I don't think that there have actually been studies of that type. In terms of separating out the stress from actual peculiar things to space or microgravity and their impact on the immune system, that's definitely been a question that's been looked at in the multiple studies that have been done on astronauts and stress and their immune function, but there just haven't been any conclusive answers. Is this related to stress? Definitely when a flight goes bad or it's more stressful, they have increased immunosuppression. But we don't know because even the flights that are completely smooth, they also experience the same immunosuppression. Mice also experience the same immunosuppression as humans do. So again, dissatisfying answer. I don't know how to separate those two things. And your second question about the hyperbaric conditions. So the studies that are done, so I'm a scuba diver, and I know some of you I know some people at the Institute are scuba divers, and what you learn is that at the surface, we're at 1 atmosphere, and so for about every 33 feet you go, you add an additional atmosphere. So at 33 feet, you're at 2 atmospheres, 66 feet, you're at 3 atmospheres. And the studies that are often done looking at the effect of hyperbaric conditions on immune function take their model organisms down to 90 feet, which is about 4 atmospheres. So that's the point where you see that you switch on the immune activity. And some of it is positive, like when people have gas gangrene in their legs, or so like they have infections in their legs, they actually put them in hyperbaric chambers. But it can also be a bad thing because there's some suppression of inflammatory cytokines. And so this is a relatively new area of study again, but I think that it's something that's interesting and should be considered when we're thinking about space. Yes?
I had a question about the compromised immune function in space. What efforts are made to untangle the causative factor, gravity versus radiation versus God knows what?
So like I said before, there have been some efforts. I'm not sure what particular efforts have been made. I think that that is extremely important, but I just think that people don't know yet, and whatever methods that they have done to disentangle those different impacts haven't yielded any results. But definitely stress is a large factor. So if we could get a stress-free environment for long-term space travelers, that would probably be better, or if they could adapt to that level of stress.
Two things. If a person reactivated a virus while they were in space travel, could they then give that virus to other people? And the second one is, what's the percentage of airborne microbes and viruses to touch contact?
On surface, right? Is that what you're asking?
Well, either place.
Okay.
Or both places. I was thinking of space travel, but also here is interesting too.
So for the first question, the answer is definitely. And a lot of organisms, a lot of viruses live in our bodies that we still have them. So herpes is a great example. Chickenpox is a herpes virus, and then also herpes simplex 1 and 2. They live in your nerve cells, and when you're under stress, they come out, and you see that in the form of cold sores on your face. And you actually are making the virus, and when you have those sores, your cells are bursting and releasing the viruses, which are infectious to other people. And so definitely, when you reactivate viruses, you are, in most cases, again, infectious to others. And then your second question about surfaces versus air. So in microgravity, you're not going to get a lot of settling. The settling is going to be primarily random. Most of the organisms that you see are going to be floating around in the air, like the astronauts, because of the microgravitational conditions. I think that one of the great opportunities that you could have is developing surfaces that actually resist microbial colonization in the spacecraft. So there's a product called Microbiome that's used in toilet paper dispensers in bathrooms, and it actually resists microbial growth. And that could be an option. But currently, the big problem is with the microbes in the air because you're breathing them in, they don't settle. And interestingly, actually, that lack of settling is one of the things that contributes to the changes in microbial physiology in space, is that normally when you have organisms in culture, they settle to the bottom and they have something called quorum sensing, which they can sense how many organisms are surrounding them. But when they're in microgravitational conditions, they're all floating around, they don't have that same quorum sensing going on, so they don't know how many organisms are around them, which contributes to their changing their growth, and their growth being different than what you would see on Earth. So.
Caitlin, we have a question from online.
Oh, great.
So the question is, we already know that as fact that some Earth bacteria flourish at Martian atmospheric pressure levels. Would we have to look ahead and see how bacteria related to humans would change in those conditions before colonizing that world?
Yeah, I think absolutely. When we think about colonizing any place, we have to think about the things that we're bringing along with us. You know, we have evolved very distinctly for life on Earth, and we've evolved alongside the organisms that live here with us on Earth to respond to earthly things. If we were ever going to go to Mars and colonize Mars, we have to think about what happens on Mars. What are the conditions of Mars? What does the pressure change do to the organisms on Mars? And I think that to some extent, we can't plan for that. That's going to be something that happens once we get there and once we establish human populations in a different place. The organisms that we bring with us are going to select out for the ones that grow better or grow worse. And they're not doing that with us in mind. They're doing it as a process of natural selection. And so, which is why I believe very strongly that we need to bring with us wherever we go in space, if we're going to recolonize, a library of organisms that we can reseed so we have some control over the natural selection that goes on inside of us.
All right. Okay, one last question.
Are there any gender-related issues of microbial diseases, like urinary tract infections for females, for example?
So the urinary tract infections
and vaginitis and things like that.
Yeah, so the urinary tract infections that I read about were actually in men, not women. But certainly, women have a complement of organisms that is different from men, because in the vaginal canal in particular. And I think one big gender issue when we think about colonizing another place or being in a spacecraft long term is we're going to want to make a next generation of people. And we have to do that by reproducing. And one of the really important things that happens during birth is that the baby goes through the vaginal canal. And in that process, they get covered in the organisms from their mother. And that plays a key role in how they're colonized, because babies are in the womb completely sterile. And when you take them out via C-section, you actually see a dramatic difference in the microbiome when compared to vaginally delivered babies. And so we have to think about, in space, how are we going to have babies? Is it possible? And if we do, how are we going to make sure that they have a microbiome that's functional and provides all the benefits that we see in children here on Earth? We have time for a few more.
Do we have any more questions? Yeah.
Yeah, a lot of the studies you mentioned up here, you talked about the Apollo program and the space shuttle, which were relatively short. What about the ISS? What kind of data are you getting from that?
Nope, no data from the ISS so far. Yeah. So here, I'll pull up my references. So Pearson is the guy who's done a lot of these studies in the spacecraft. And in 2011, he wrote a summary of his findings from the space shuttle programs. And those are the studies on space epidemiology that we have. ISS, there's similar microbiological protocols, there's similar microbiological testing, but it hasn't been assembled and published, and conclusions haven't been drawn. And I think that that would be a really excellent source of data, because people are up there much longer than they would be in a space shuttle.
And you've got other nationalities up there all the time, too.
Oh yeah, you get a bunch of mixing. And I think that what Jill mentioned earlier, that would be a great way to see if there's homogeneity in people who've been living in space for a long time.
You mentioned there was a difference between vegetarians and carnivores, but you didn't say which was better.
So I think that what I want everyone to take away from here is there's no better, there's no worse, there's only adaptation. There's only what works for that person and their diet and their environment. You can't say, oh, you know, Prevotella is better than Bacteroides in your intestine, right? It's only, does that help you digest the best? And if you're eating plants, you're going to select for the organisms that are better at digesting, you know, cellulose. If you eat meat, you're going to have organisms that are better at digesting proteins or things that are found in meat. So I think that we need to get away from this idea of better organism, worse organism, but rather what works.
This is kind of a long-term question, but
Yeah,
I'm just curious if anyone is looking even farther down the line at what would happen several generations later for humans living on Mars or living on planets around distant stars that wanted to then return to Earth to see planet Earth. It seems like you'd have the same whole issue in reverse.
Yeah, so there is a whole complement of issues related to bringing astronauts from, you know, microgravity, increased gravity, decreased gravity back to Earth, and what happens to them. And I think that you see that people come back and it takes them a while to recover from the immunosuppression. I'm not an astronomer, I'm a biologist, and so I feel like my answer to your question is going to be limited to what I think will happen to the microbiome. And I think that you're going to see people coming back And they're going to have some problems. I think that one of the big problems that we're going to see if people are in space for long term is they're going to have the decreased diversity of their gut microbiome, perhaps, which on Earth will lead to this constellation of irritable bowel disorders, where they have trouble digesting food. They've been living on a spacecraft diet. They come back, they want to eat like their tasty Mexican food. That might be a problem. And I think that this goes alongside with what I said about developing probiotic therapies that actually work, so that they can come back to Earth, they can start eating the yogurt or drinking the yogurt drink or taking the pill, and it actually makes an appreciable difference to their microbiome. And that's something we definitely need to look at.
So, Caitlin, just a point of clarification with respect to the epidemiology and the ISS. Is it the case that there are no data, or that there have not yet been any analyses?
No analyses. There's definitely data. People are routinely swabbed in their nose for the methicillin-resistant Staphylococcus aureus, which is like the really serious hospital-associated infection where people get the little, what looks like a spider bite that turns out to be a very resistant skin pathogen. So they swab people for MSRA routinely before they put them onto any sort of space mission. They also look at their health before and after. They do blood samples. But in space itself, there's a lot of limitations in what you can do because of the limited medical personnel there. And so the data we have wouldn't necessarily be complete enough to do an epidemiological study. And I would say that there are definitely limitations in the space shuttle missions as well, because all of the data is taken before or after. But as far as I know, there have been no analyses made of the data collected from the ISS. That is just routine spacecraft associated data, but there have been analyses of the data collected from the space shuttle missions.
But is there a community that's arguing for more epidemiological studies?
I don't think that there is a community arguing for it. There is some work done. The major papers are done by one person and a few colleagues, and he wrote a handful of papers about the space shuttle. And then there was the paper that was published in the 1970s about the Apollo missions. And beyond that, I think that the majority of the push is for keeping astronauts healthy in space. It's not about how do we take them just beyond that basic level of functioning and bring them into actually achieving those benefits that can be conferred by the microbiome. Does that answer your question? Okay, perfect.
So you had a lot of fun, popular science fiction references of the Captain Kirk and the like. Just to let you know, the question is, do you get in the A arc, the B arc, or the C arc? And you make sure you have the telephone sanitizers with you. That's one thing. Douglas Adams only died 100 miles away from you, by the way, in Santa Barbara.
Really?
Yeah. So, if you go back and watch The Hitchhiker's Guide, and, because that's actually what wiped out one particular population, but I won't give away the thing, the storyline.
Yeah, there's something about space that captures our imagination. There's something about bacteria that captures our imagination. And I think that space and bacteria can be an area that generates a lot of popular interest because people like to know what's living inside of them.
Yeah, you need a VR reference.
I'll make a note of that, okay?
A couple of comments. One, NASA does now have a new program, Microbial Observatory, that's funded to do exactly what we've been talking about, the changes in the biome, and Gene Lab, which is another new program looking at the complete genome analysis from organisms, including humans, that are flown in space for an appreciable period of time. Last comment is the station walls, like Mir before it, are covered with microbes.
Oh, yeah.
Biofilms. And biofilms are a huge area of problems, both here and in space. And there's programs and grantees that are now investigating actively the effects of spaceflight on these quorum sensing biofilms.
Definitely. So that's what I was talking about when you have to design your space environment alongside with the microbial invasions that you're planning for. And the nice thing about surfaces, though, is you can pull out your Clorox wipe and you can wipe it down. The only way you can do that in space to the air is by filtration. And so there have been advances in filtration technology and advances in sanitizing the surfaces that are really important. I think both of those programs are going to be vital, indispensable to a long-term space travel mission.
Is there any evidence or occurrences of microbes evolving in space and come back in a form they haven't been seen before? Is there anything? Is there a danger in that ever happening?
Yeah, definitely. So definitely. So when you're in space, you're exposed to a level of radiation that's much higher than on Earth. And when you have radiation, you're going to get the genetic mutations. And some of those mutations are lethal. Some of them are actually beneficial. And there's a study done where they sent a model organism into space and looked at the mutations that happen in a particular plasmid, so a circular piece of DNA that they put into their model organism, and looked what happened to that particular plasmid. And certainly there was a dramatic change in the number of mutations. And the Salmonella example, it didn't come back as a different species. And I think that what you mean by coming back in a different form, maybe not a different species entirely. It doesn't, become a multicellular organism. It doesn't become a frog. But it definitely has distinct changes in the genome that can be very harmful to human, that happen when it's exposed to microgravitational conditions. So, definitely.
Has there been any effort to develop antimicrobial coatings, catalytic coatings, for the surfaces of spacecraft to mitigate against airborne bacteria? Even though they don't settle, they'll probably have a finite, they will contact every surface eventually, statistically. So what's been done in that area?
I think our audience member in the back would have some good information about that.
The answer is yes.
Yeah.
Yes.
Okay. In J.R. Roth's novels, you can get it 50 years from now. Everybody can get it.
We can get a lot of things 50 years from now that we'd all want today.
Okay, last two.
Okay. I believe in hospitals they use UV light to cut down organisms. Has that been tried in the International Space Station or anything in orbit?
Yeah, so I know that they've done some experiments recently with microorganisms on the outside, deliberately exposing them to hard vacuum, and that things as large as water bears were able to be revived in some cases. And so it occurs to me that when you're doing, like you're in the ISS and you're doing an EVA, you've got microorganisms on the outside of the spacesuit. You go outside, you expose them to the hard vacuum. Some of them maybe cling to the side of the space station and are there until the next time you go out and rub, and then you come back inside, and now you have mutant space bacteria, right? And so my question is how unpredictable is this stuff?
Is that it shows, it's a way of us looking at what happens to organisms in the environments that you guys are most interested in. What happens to evolution if they're exposed to that hard vacuum? And that's where those that microbial library, the facilities on board a spacecraft to do actually wet lab microbiology research is going to come in handy, because you can take samples from the spacecraft that you're traveling in that can tell you information about what you might have when you arrive in place, and send back important information to Earth about how the organisms that are here And I think that, as with all of these questions, there are just so many wonderful frontiers to be explored with all the microcosmos that we carry with us. And I'm really excited about what's going to come next. And I think that a lot of people are very excited about what's going to come next in this area.
Okay, Caitlin, we have a especially microbially, I don't know, infected. I've been carrying it around for a while, and I won't talk much about that. Please join me in thanking Caitlin for a great talk.